everyone, and welcome to our cleaning validation webinar. I'm Michelle Christian from International Products, and I handle the sales and the marketing for the company. Perhaps I've sent you a sample or spoken with you about our products, but I'd like to welcome you to our webinar. I'll begin with a brief overview of our company. International Products manufactures specialty cleaners and P80 rubber assembly lubricants. Our manufacturing facility is headquartered in New Jersey, and we have worldwide distribution. Our cleaners are primarily used in industries with a diverse range of hard surfaces that are required to be critically clean. We're often asked to supply validation methods, and we thought this webinar would be beneficial. We will review the most common cleaning validation techniques used. Now, in an effort to save time and to provide you with as much information as possible, please submit your questions during the webinar. On your screen, you'll find a field where you can type your question and submit it online. At the conclusion of the webinar, we will answer all of your questions. I'd like to introduce you to Thomas McGuckin, our VP of Research. Tom has been with the company for over 12 years, and he handles all aspects of research and product development.
Thanks, Michelle. And as Michelle mentioned, today we're going to talk about cleaning validation and cleaning validation techniques. The problem, though, is that cleaning validation means something different to almost every company out there. Depending on the company and the industry that they are in, as well as what's being cleaned and how they clean it and their specific customer requirements, cleaning validation can take on a broad number of definitions. We feel that these companies can be classified into two broad categories, and they are those who are regulated by the FDA and those who are not. In general, it means that the detection limits for both of these groups are vastly different. Typically, FDA-regulated companies have very low detection limits. They need to know what the parts per million or even part per billion concentration of cleaners are versus non-FDA-regulated industries, and their concentration requirements are usually in the percent. Also, we've noticed that depending on the company who performed the cleaning validation, what they actually analyze can vary. Some are concerned with the specific concentration of the cleaner. Others are concerned with the lack of cleaner. And what I mean by that is they will check rinse tanks or rinse water and verify that there is no cleaning solutions in those tanks. And finally, still others aren't primarily concerned with the cleaner concentration at all in either the cleaner baths or the rinse water baths. Their primary concern is the lack of soil, which hopefully is on their part that they are cleaning. So there's a lot of inspection variables that need to be considered. The cleaner bath and what the concentration is of the cleaner, the rinse water bath, and hopefully a low or no concentration of the cleaner. The part that's being inspected is analyzed for either cleaner residue or soil. And also, even the empty tanks and equipment inspection. Before or after a cleaning campaign, the inside of this equipment is sometimes analyzed, usually through a swab technique, to make sure that they are clean sufficiently. So the technique variables, the validation technique variables differ significantly. Essentially, it's qualitative versus quantitative techniques. A basic visual inspection of parts is sufficient for some customers, whereas others need to know the exact concentration of the cleaner in different solutions. So we've assembled 10 cleaning validation techniques that span a lot of different industries, and we like to discuss some of them. We like to discuss them and describe essentially what they are, some of their benefits, and some of their drawbacks. The most basic technique is just visual inspection of the part. It's a way to check for soil And although it is very basic, it is also very reliable. It can be done systematically using what's called the water break rinse test. In this test, the part that is just cleaned and hopefully rinsed properly is removed from the rinse water, and as it's removed, the clean rinse water should sheet off the part uniformly. If that's the case, then the part can be considered clean. However, if water droplets appear anywhere on the part, that's a sign that the part needs to be cleaned again. This type of result is immediate, it can be done tank side, and it's very low cost. It only requires the operator's time. The results are qualitative, which are sufficient for some but not others, and in most cases it only detects gross contaminants. Another drawback is this test is subjective and it is operator specific. Another test is the foam test, and this is predominantly done by evaluating the condition of rinse tanks. And the main purpose of the foam test is to check for cleaner carryover in rinse tanks. Rinse tanks typically have mild agitation, usually in the form of cascades, and this is enough turbulence to create some foam. And most traditional hard surface cleaners do generate foam, and we have found that if the foam does not break and sits on the top of the rinse tank, then you can assume the concentration is about 0.25% or more. Anything below that is not necessarily accurate. Similar to the visual test, the results are very quick, minimal to no cost, just operator evaluation. However, the drawbacks with the foam test are soils and sometimes hard water may interfere with the results. Hard water will certainly reduce the foam, and soils could either reduce or contribute to the foam. And again, it is a subjective test. It is operator specific. So one operator may consider the foam significant and it's time to change the cleaner baths, while another may think differently. Another cleaning validation technique is titrations. This is a little bit more analytical than the previous two, and its purpose is to check for the cleaner concentration. Titrations are quantitative, and we have found that the detection limits are about at 0.1%. Titrations are very quick and easy to do. However, you do need a calibration curve in order to determine what the concentration is. Calibration curves should be available from the cleaner manufacturer. However, they are easy to develop. Titrations work best on alkaline or acid cleaners. They don't work well on neutral cleaners. They are low cost to implement. However, they're ideally suited for lab work. It's not something that can be done easily right by the tank, simply because you need glassware, reagents, perhaps solvents, and indicators. Some soils may interfere with titrations, which is a drawback. And again, the use of solvents is considered a drawback because of handling and waste disposal. Just like titrations, the refractive index is another method that provides quantitative results. It again checks for cleaner concentration, typically in the cleaning baths or the rinse tanks. The results are fast. They're even faster than titrations. Refractive index requires minimal technique, and another positive aspect of it, it can be done right at the tank. Like I said, the results are quantitative, but you can't achieve the same limits as you can with titrations. We found that about 0.25% is as low as you can get with refractometers. And similar to titrations, a calibration curve is needed. But again, these can be provided by cleaner manufacturers. There are no disposal costs with the refractometer or the refractive index tests. You just simply apply a small quantity of the cleaning solution on the refractometer and take a reading. So the costs are minimal. The handheld refractometers are fairly reasonable in price, and there are digital versions out there as well. However, some of the drawbacks are foam, excess soils, and cloudy solutions will interfere with the reading. And then finally, you do need some sufficient lighting in order to get an accurate reading, and that may not always be available in manufacturing plants. Conductivity is a very popular cleaning validation technique. Similar to the previous ones, it checks for cleaner concentration, and it gives results very quickly. It does require a minimal amount of technique, and you do need a calibration curve. It is also quantitative. However, the detection limits are down to about 50 parts per million. The costs are low to moderate, depending on what type of instrument is purchased. The handheld conductivity meters, again, are fairly reasonable in price, and there are versions out there that are permanently part of the tank, and you can get readings on time, real time, and continuously. Conductivity validation techniques are popular with both FDA and non-FDA regulated companies. We find that FDA-regulated companies may use this technique as a primary evaluation of the cleaning concentration, and then if this concentration exceeds specifications or control limits that they've established, then they may refer to another more analytical cleaning validation technique. There are some drawbacks with conductivity, however. You do need higher quality water, ideally distilled or deionized. If you use tap water and the tap water is fairly hard and you use a low concentration of cleaner, then your results will not be that accurate. Also, conductivity is temperature dependent. Most meters, all conductivity meters have an automatic temperature compensation factor. So these factors must be employed in order to ensure that the temperature is not giving you erroneous results. And similar to titrations, conductivity is not really suited for pH neutral cleaners. The next cleaning validation technique is called the Methylene Blue test. Similar to the other ones, this also checks for cleaner concentration, but it's predominantly used on rinse tanks to check for the lack of cleaner concentration. It is semi-quantitative. The detection limits are somewhere between 10 to 50 ppms. We found that to be true for most of international products cleaners. Minimal cost is needed in order to set this test up. And another benefit is the results can be obtained rather quickly. It is somewhat subjective, and what's unique about this test is it is ingredient specific. So it won't work on all cleaners. You do need to know some information with regards to the cleaner formulation in order to be ensured that this will work. A drawback with this test is you do need solvents, specifically chloroform or N-propyl bromide. So handling and concern is an issue. And just like the refractive index test, you do need sufficient lighting, so it may not be ideally suited for tank side evaluations. The Methylene Blue test can be enhanced by the use of a UV visible spectrophotometer. By using a UV visible spectrophotometer, the detection limit is greatly enhanced with a detection limit of less than 10 parts per million. So this technique is commonly used by FDA-regulated companies. It eliminates the subjectivity, and it is one of the least expensive choices for companies that need to have that low of a detection level. You do need the lab equipment. You do need to purchase the UV visible spectrophotometer and a centrifuge as well. The calibration curve needs to be developed, which takes some time. Results take a little bit longer than with the manual method, particularly if you're developing the calibration curve for the first time. However, having said that, results are available within 30 minutes to an hour. And again, you do need to work with solvents, but the volume of those solvents are reduced significantly if you're using the analytical equipment. The next technique is the Backlight Clean validation technique. This technique can be used for several different reasons. It can be used to check for soil or for cleaner residue. And black lights are typically available at two different wavelengths, either 254 or 366 nanometers. When evaluating parts, users are typically looking for soil or cleaner residues. Oftentimes, though, black lights are used when new tanks or equipment is purchased and the coverage wants to be evaluated. These results are qualitative, and the costs are low to moderate depending on the black light that's purchased. You do need a dark area to evaluate the parts. At times, cleaner residue or the soil may not be fluorescent, which is required to see on the parts. But that's not necessarily a problem because fluorescent pigments can be purchased and they work at very low concentrations. So a small amount of a pigment can be added to your cleaner solution, and then you will be able to check for the coverage inside tanks or any residues remaining on parts. There are possible hazards with black light, specifically skin and eye exposure. The next validation technique is total organic carbon. And the purpose of this technique is to check for organic carbon. Typically, this is done in rinse water, either from tanks or water from the parts. It provides quantitative results, very low detection limits in the parts per million. The results are obtained pretty quickly, and this is used predominantly by FDA-regulated customers. This technique is not ingredient-specific, and that means if you do get a high result, you don't necessarily know what the source is of that high result. It could be cleaner, soil, or contaminants. These instruments are higher cost than everything we've talked about thus far. Ideally, they're suited for laboratory work, and these only work on water-based cleaners. Oftentimes, solvents are used as a final rinse solution. If this is the case, then total organic carbon is not an appropriate cleaning validation technique. The last cleaning validation technique I'd like to talk about are gas, liquid, and ion chromatography. These instruments check for cleaner concentrations. Typically, what's analyzed is rinse water or swab. They're the most versatile techniques available, providing quantitative results in the parts per million or even parts per billion concentrations. They do evaluate specific cleaner ingredients, and once the instruments are set up, results are available in several to many minutes. These are run predominantly by FDA-regulated companies. However, once a method is developed, the instruments can be set up and left alone. They run automatically. The costs are relatively high for the initial purchase and also the upkeep. High-purity solvents, reagents, and high-purity water are necessary in order to have these run properly. And typically, most companies have a technician, full-time technician, running these instruments. It does require method development before samples can be run, and this can be quite timely as well. And samples need to be prepared before running them. This chromatogram is a typical IC analysis of several anions. This is a 7-anion standard, and these are the results possible with ion chromatography. The 7 anions that you see listed here range in concentrations from 1 part per million to 20 parts per million. So this is the type of analysis that is possible with gas, liquid, or ion chromatography. Okay, this concludes my part of the webinar. I'm going to hand it back over to Michelle for additional information.
All right, thank you, Tom. Thank you so much. Also, I want to let you know that I will be sending you a chart that outlines the cleaning validation techniques that we discussed. Okay. So, and included in that will be our contact information if you have any additional questions or if you need cleaner samples. All right, so we'll go ahead. Tom will answer a question. We have a question about riboflavin.
Thanks, Michelle. Yeah, one of the questions that we received is there a reference for riboflavin concentration or an accepted industry standard? And I'm assuming this question pertains to the Backlight method because riboflavin is usually added to cleaner solutions, and we can see the fluorescence of the riboflavin as it is mixed in tanks or on parts. We have found that the riboflavin does fluoresce. However, you need a slightly higher concentration. We found that it works quite well between 10 and 25 parts per million. Personally, we like more of the synthetic water-soluble fluorescent pigments, and one of them in particular we like to work with works down to 1 part per million in the cleaner solution. And these are available from companies that just specialize in these pigments. And if anybody needs any other information, we can certainly provide the manufacturer and the product, although I think it is listed on our website.
All right, Tom, we have another question about the pH level.
Okay, and it's basically what do we consider a neutral cleaner? Is it between pH 6 and 8? And yes, we consider anything between 6 and 8 a neutral cleaner. Anything above a pH of 8 is considered alkaline, and below pH of 6 is considered an acid cleaner.
All right, there's a question regarding TOC and inline.
Okay, yeah, I can see it. Do companies use online TOC in their rinsate lines? Most companies that use TOC seem to use them as standalone procedures. In other words, they take a sample, they run it off their line, they run a TOC analysis. I haven't heard of any companies that do them online. I assume that technology is out there, but we haven't come across any that do it online.
All right. Now here's some common industrial limits for residual detergents. That's another question.
We find that it actually depends on, I guess, the ingredients of the detergent. Most companies that contact us and want to know what limits they get down, what limits are acceptable, typically they come up with their own limits based on the cleaning campaign and what follows their manufacturing or what follows after the cleaning campaign. It seems like when we have a new customer on board, they ask us for a lot of the toxicity data of our cleaners. They ask us for suggestions for the validation methods, and they want to know how low each of those methods can go. And also they usually ask for data such as cytotoxicity tests or inhibitory residue tests because they typically provide some toxicity limits. But in general, I mean, if someone's looking for an absolute limit, I would say anywhere below 10 ppms, although I don't know of any companies that actually use an absolute limit. It seems like they take all the data that we provide them and they determine their own thresholds.
All right. Now we have a question. How do you set ARL for TOC? And also, how do you correct acceptance limits with carbon factor?
You know, to be honest, I'm not really sure how to answer that. It seems like it would depend mostly on the specific instruments. We found that TOC is used, but to be honest, it doesn't seem to be that popular with regards to validation. We found that a lot of companies use it as a somewhat fast analysis, but yeah, to be honest, I don't know how to specifically address that question. That seems like it would be better suited for the TOC manufacturer.
All right, and we're still taking questions, so if you have any questions, please feel free to send them over. Also, just to give you a little background about our company, we do have a lab where we can set up validation methods for you. If you have a particular soil that you may have a problem removing, or if you are looking, I know that a lot of companies now are overstaffed where they're not able to do a lot of this testing in-house. Please feel free to contact us. You can send us coupons or samples of the type of soil and just give us some detailed information on what it is that you're cleaning, what problems that you have. We have a line of detergents that come with different validation methods. I'll be sure to send that with you along with the validation chart that has the different methods. Did you have any other common questions that we may get dealing with validation?
Well, a lot of times we get people who look at our website and say, you know, we want to try a specific cleaner, and we've been using a certain way to validate or to check the concentration. And one of the most common ways is conductivity. We come across that quite a bit, and we do have conductivity charts available because that seems to be a very common question for our alkaline and our acid cleaners. And I believe they're on our website, but if not, we can certainly email or fax them to whomever is necessary.
All right. Well, let's see. Okay, here's a question. Is residue on evaporation applicable?
I'm assuming they mean as a validation technique, and certainly that can be appropriate. Similar to TOC, though, you're not going to know what that residue is. So you won't necessarily know if it's a contaminant or if it's a cleaner residue or if there's something in your water that shouldn't be there. So, yes, it can be used. To be honest, it's not that popular, though. I've come across it one or two times. Usually some people, when they're first setting up their validation technique with a new cleaner, they may use that just to make sure that their rinse steps are adequate. And then once they determine that the rinsing is appropriate, then sometimes they switch over to HPLC or another analytical method.
All right. All right, so does anyone else have any other questions? We have a little more time if you want to send something over. All right, so I guess we will be wrapping this up, and I want to send out a thanks to everyone who took the time out to register and to join us. I hope that we were able to inform you. Again, if you have any questions, please feel free to send them to us, and we will answer them. Also, this webinar will be available for download for viewing by the end of the day. So we'll have that available for you as well. Thank you very much.